Hi, here is our notes on proteins. This is our last macromolecule. Okay, so proteins, we saved the biggest and best for last, I guess. They are the most diverse group of our macromolecules. They are made up of monomers called amino acids. So a protein is a polymer, and its monomer is an amino acid. There are 20 different amino acids. And scientists think that amino acids were among the first molecules on Earth, probably in the early oceans where before life even began, it was teeming with these structures called amino acids. We typically think of proteins as the things that we get from the meat that we eat, or maybe eggs or cheese or beans or nuts. But we're going to learn a little bit more about proteins today. So, first of all, proteins are way too diverse to be able to list them all. So they are broken down into different categories based on their functions or their properties. So we have enzymes. All enzymes are proteins that help to speed up chemical reactions. We have our defense proteins or our immunoglobulins. These are antibodies. These are the things that are going to help to recognize those foreign invaders that get into our body and start the process of getting rid of them. Okay, so that you don't get sick. We have transport proteins. There's ones that circulate throughout our body and transport things around our body. And then there's also membrane proteins that transport different substances into and out of our cell membranes. And we'll learn a little bit more about those when we talk about membranes in the next unit. There are support proteins that are consistent of fibers, and they usually play structural roles like the matrix of your skin, your hair, your nails, and other parts like that. We have motion proteins that are your muscles, your actin and myosin. We have regulatory proteins. There's osmotic regulatory proteins that help to regulate the flow of water. There's gene regulatory proteins that are going to turn on and off different parts of the DNA. And we'll get to learn a little bit more about how that happens in later units. And there's also hormones, which are messengers and receptors that will activate certain parts of our DNA or go into our cells and start reactions. And then, of course, we have the storage proteins, which bind ions like calcium and iron, essential things that we need. So every amino acid has the same basic structure. If you were part of our synchronous time, you got to see me describe this structure, but I'll go over it again. So in the middle, we have this central carbon always, and off of that central carbon is a hydrogen. Okay. To the left side is an amine group. So we got an NH3, which has a slight positive charge. And then to the right, we have a carboxyl group. It's just COO, and that has a slight negative charge. Okay. And then we also have an R group, which is the variable group. This is where the basis of the property of each amino acid is going to be different. So all of these other parts are the same, but this R group, this group down here that changes, is going to give it all of its functions and properties. Okay, so there are those 20 amino acids are grouped into 5 different categories. And those 5 categories are going to, with their properties, are going to determine how this protein is shaped and its function and what it does. So we have nonpolar proteins whose variable R group usually has a CH2 or CH3 in it. We have polar uncharged amino acids whose R groups usually have an oxygen or an OH in them. We do have charged groups that have those R groups or variable groups with acids or bases that easily dissociate. We have the aromatic groups of amino acids, which are also nonpolar, and those have carbon rings with like alternating single to double bonds. And then we have our special or unique amino acids like methionine, which is usually the start of all of the protein chains. If you remember from your general bio class, you would have started with DNA that was TAC, which translated to mRNA, which was AUG. And then if you looked that up on an amino acid chart, you found methionine. Okay. So do you need to memorize all of these? Not at all. But you do need to know that there are different kinds of amino acids, and their interactions with each other are going to be different because of their charge, no charge, polar, nonpolar, and the different structures that they have. Okay, so how do we make a protein? Well, we make a protein by putting together peptide bonds and by putting together amino acids with peptide bonds. And guess what? Just like we've learned in all of the other ones, this is a process called dehydration. We're going to remove an H from the amine group and an OH from the carboxyl group of the other amino acid. We'll have a water that forms, and then a peptide bond will form between the carbon and the nitrogen of those two amino acids. Once we get a chain of these together, we call that a polypeptide. Okay. When we're talking about proteins, though, there's 4 different levels of protein structure. Those are primary, secondary, tertiary, and quaternary. This isn't the best image, but we'll go through what each one is. So the primary structure of a protein is just literally the chain of peptides, amino acids that are connected together. There are so many different ways, and they're so diverse because we have 20 different amino acids. So if you had just a very simple protein, which most of them aren't very small, but if you had one that was made up of even just 10 different amino acids and you have 20 to choose from, there's so many different combinations. So you can imagine some proteins could be 1,000 or more amino acids long. So that could be a really huge protein, and there could be so many different kinds. So that's why they are so diverse and important. The secondary structure it happens from hydrogen bonds that form between nearby amino acids. Okay, so after they're connected with those peptide bonds, we get some hydrogen bonding forming, and it forms 2 kinds of structures, alpha helices and beta sheets. And again, that's just because of the hydrogen bonding. Then we have our third level or tertiary structure. This is hydrogen bonding between those variable groups. So not just between the amino acids, but between the variable groups. And then we have some hydrophobic exclusion from water. So what scientists have found is that all of the nonpolar R groups, the amino acids with R groups that are nonpolar, are going to be folded inward. They're going to want to be on the inside, away from that water. And then we also have some ionic bonding between oppositely charged regions. So we get this nice little folded up protein at this point. Then if, and is the case in many, you'll have 2 or more of these polypeptide chains that have been folded based on their polarity and their hydrogen bonding and their R groups. You'll get these to associate together, and they form a fully functional protein. And it's like if you took a paper and somebody else took a paper and you smushed them together and you created an object. That's what the quaternary structure of a protein is. So we have a couple more things on proteins that help us to understand a little bit more about their functions. There's motifs, domains, and chaperones. So motifs are similar sections between proteins. So you might have two totally separate proteins that are made up of different amino acids, but they have a similar function. So scientists think that this could possibly be a reuse of, so this worked through evolution, and so why not keep redoing this general structure? Okay. So it doesn't mean that they're 100% identical. It just means that they have similarities, and because of those similarities, they're going to do similar functions. Then you have domains. Domains are just pieces of a larger structure that will have certain properties and help it to fold into a proper shape or a different shape. So you might always have, like, this is a domain here, and this is a domain. So we'd have different domains as part of that final protein structure. And then finally, we have the fun guys, the chaperone proteins. And this is where, it gets me to thinking. So we have proteins that help other proteins to fold properly. But in order for those to work, they had to be created themselves. So it's kind of that whole like chicken and the egg thing, which came first. So you need these chaperones to help fold certain proteins, but the chaperones themselves are proteins. So that's basically their function, is just to help proper folding. So what happens if things aren't properly folded? So an improper folding of a protein can lead to a disease or disorder like cystic fibrosis, or some of the ones that are listed in that chart. So Huntington's disease is the Huntington protein is defective. That's a really debilitating neurological disease that usually has a late onset in the 40s, the age range of the 40s. And so many people don't even know that they have the gene for it, before they have kids. And then another thing that can happen is denaturing of proteins. And denaturing of proteins is when we change the environment that the protein is in. Proteins are usually very picky. They want to have a certain pH, a certain temperature, and a certain ion concentration. If those things get out of balance, they just all fall apart. And that's called denaturing. Okay. So if it could possibly be put back together, some can, some can't, then that would be renaturization. But denaturization happens a lot more, which is a loss of biological activity because something went wrong in that environment and caused it to unfold or change its shape. The best example that most people know is if you crack an egg into a pan and you have that albumin, that egg white is clear when you open your raw egg. And then as you apply heat to it, it changes colors. And that's because those proteins are being denatured. So we're changing the shape of those proteins, and that's why we get the different color. So I hope that you had a good time learning about proteins, and I will see you in class. Thanks.